series. We will wait just a little longer so that we can give time for people to join. And then I will introduce you our speaker, and then we will start to talk. Just one brief introduction on how the seminar will go. You can ask the questions on the little button down there where you see Ask a Question. And other attendees can actually vote for your question. So if you want, you can also take the stage and ask your question. Just let me know afterwards what you prefer. And also you can leave your comments on the chat. So the talk is going to be recorded. So if you know someone which is not able to attend the talk live, we will share the link with you, and you are free to share it. Yeah, so I think if one more thing, we will have a Q&A session afterwards. We still, I think, will be able to accommodate some more people, maybe 2 or 3 people more. So if you want to join, maybe you can drop me your email and in the chat, and then I can send you the link to the Zoom room. So just feel free to let me know if you want to join us in this discussion. So I think we might just wait a couple more minutes, as we still have some people which need to join. Okay, maybe we wait until 4:05. So just a couple more minutes, and then I start. Okay, welcome to the ones who have joined since. Okay, so I'm gonna start by introducing Monica. So I'm very excited to be introducing Monica Dus today for our body-brain interactions series. So Monica finished her PhD in 2008, and she carried out her project in the Hannon lab. And during that time, she was mostly focused on studying the biology and the function of non-coding RNAs. And in particular, she studied the role of piRNAs in transposon control, and she contributed immensely to what we know today of this pathway. So since then she joined the lab of Greg Sue for her postdoc work in Cirqueball Institute at the NYU. And she started focusing on how the fly brain perceives nutrient values of foods. And so she has been working on trying to unravel what are both the molecular mechanisms and also the neuronal circuits which underlie this perception of nutritional and non-nutritional sugars. And she contributed a lot to what we know today about this, and also how these pathways are modulated by the internal state, and namely the sated and stated states. And so since then, she has her home lab now in University of Michigan, and I think she's kind of fusing her interests and backgrounds in both the genetic control of genes and of pathways, with also the modulation of feeding and physiology. So she's bringing together a lot of different fields, let's say, and she's putting that effort into understanding how or why animals overeat certain nutrients, and how this impacts the animal physiology, the brain function, how this promotes feeding intake and weight gain. And so, as I told you, she's kind of taking this multidisciplinary approach, and she's bringing fields together such as neuroscience, metabolism, epigenetics, and it's really just the way we like it. And she's using the drosophila model system to do it, which is perfect. So we want to welcome her today. It's a pleasure to have her here today with us to tell us about what she has been doing. And so I really look forward for her talk and for discussing with her. And so I give her the stage. So thank you, Monica, for being here today with us.
Hi, everybody. Thank you, Zita, for the introduction.
So let me turn off
the mic. Yeah, great. We had some feedback. So today I'm going to tell you about a project that we've been working on in the lab for the last few years. So whether you are, it's not advancing.
Could it just be
maybe like this?
No. Oh yeah, it's working.
Okay, great. So whether you're a fruit fly or a human, or my dog Cupcake, which I think you can see her in the bag, our chemosensory system, the taste, the smell, really unlocks a lot of the life pleasure. For me, it's a cupcake or your favorite chocolate cake, your favorite cheese. And among the chemosensory senses, the sense of taste is what allows us to perceive qualities in foods such as sweetness, saltiness, creaminess, and savory. And it happens that we really love these qualities in foods, and studies suggest that most of our food choices are driven by how much we like the food, which depends a lot from smell and taste. And actually, our food preference for this type of taste qualities like sweetness and creaminess are innate. And you don't have to trust me on zombies. You can look at the facial expressions of kids eating vegetables or kids eating sugar to immediately see how early this preference develop and how also innate they are. But it turns out that although most of our preferences are innate, the reason why we like things like sweetness or saltiness is because the chemoreceptors in the mouth, they're wired to brain areas that give them a positive or negative balance. And people think that this balance is where, due to our ancestors' evolutionary associations with things that give them a lot of calories or energy, or things that kill them, like a lot of plants will have poisonous substances that tend to be bitter. And so over evolutionary time, we learn to avoid bitter and pick sweet things that also give us energy. But it turns out that even if our preferences are innate, they also change by the environment. And so some of this are due to exposure. For example, the first time you drank coffee, you probably didn't like it, but with time you develop a preference for it. And our taste system changes throughout development, it changes with sickness, with disease. But it turns out it also changes depending on the composition of the diet we eat. And so this is really fascinating. And a lot of different labs that I've listed here in different names over different model organisms have shown over the last 3 decades that the levels of fat, salt, sugar, and even better in our diet can reshape our taste sensation and our food preferences. And now I find this really, really fascinating, especially thinking about what I told you before, that studies showed that most of our food choices are based on taste, because the way we procure food is very different than our ancestors, where they would hunt and gather for food or eventually grow their own food. But death for starvation and was a real possibility in our ancestor, whereas today we go to the grocery store to hunt for our food. And the type of food we eat at the grocery store, that it's available at the grocery store, is quite different than the type of food our ancestor grew up. And in fact, this food tends to be very processed, which means that things like salt, sugar, and fat are added to it, both to preserve it, to hide the bad flavors from preservative, and also to appeal to our innate preference for this food. And we know that in humans, from a study from last year, but from a lot of other probably personal experiences, that when we eat processed foods in our diet, we tend to actually eat more calories per day here in the blue line than when we eat unprocessed food. And over time, these extra calories can result in weight gain. And over a lot of time, this weight gain can give rise to things like obesity and metabolic disease. So one of the questions we have in the lab is whether this modern diet, this Western diet that has a lot of processed food out of convenience, can reshape taste sensation and change feeding behavior, and particularly reshape the sensation of those qualities that we are innately attracted to. And so this is sort of a field, like Zita said, that has many different subjects in it. And some people call it the sensory neuroscience of nutrition and health. And this is what my lab studies. So I told you earlier that a lot of people have shown that diet composition can affect taste and food preferences. But really, we do not know how this happens. We do not know how we go from different nutrients content in diet to changes in the taste system. Does it happen in the taste buds? Does it happen in the way that tastes are processed, the flavors are processed in the brain? And then how did this changes in sensitivity, intensity, or processing actually result in something as complex as a change in food preference and food intake? And so in my lab, we studied this question, like Zita said, by studying the fruit fly Drosophila melanogaster. And that's for a couple of reasons. First, the taste system in mammals, it's quite complicated, but in flies, it's quite simple. So although it's organized along similar principles, where we have chemoreceptors in the oral cavity, which in the fly is the labellum at the tip of the proboscis, that project eventually into the brain where the taste and flavors are processed. Now, in flies, this happens in an area of the brain called the subesophageal zone. And then there's also conserved neurochemistry of taste processing and reward between brains and mammals. And in flies, we have this really powerful genetic and neuroscience tools that we can use to uncouple dietary exposure with things from things like weight gain or do other manipulations that I'll show you shortly. So, of course, we can give fruit flies chocolate or a cupcake to eat. So instead, we use a model of a high sugar diet as a proxy for processed food. And this model was developed by Laura Musselman when she was a postdoc. And essentially what it does is adding extra sugar to the flies diet. Incidentally, this is interesting because sugar is we have a lot of added sugar in our food. And in fact, 80% of grocery store products contain added sugar, including things like granola and yogurt, which we usually think as healthy. But there aren't as many studies of how sucrose or sugar changes taste compared to things like fat or salt, for example. And so if we think about the sweetness of the diets that we're giving fruit flies here, 5% is close to the sweetness of strawberry, and 30% is close to the sweetness of chocolate chip cookies. And these are not the sweetest things in our diet. And in the United States, most of the kids' cereals actually have really exorbitant levels of sugar, up to 60%. So I'm going to summarize the next few slides because the paper is already published, but this was the work of a graduate student that just recently graduated, Christina May, and then another graduate student, Anomit Vaziri, and it was published last year. But what Christina and Anomit found is that when we give flies a high sugar diet and we measure their feeding behaviors over time, on 5% here in the light pink line, orange line, the feeding is stable. But then as they're given a high sugar diet, they escalate their feeding behavior, kind of similar to the graph I showed you for humans. And then as in the graph for humans, if you look now at their levels of fat or triglycerides in their bodies, with time compared to the control diet at day 0, they increase. And so the flies are developing obesity. So I'm actually not going to get into it today, but it turns out that on a high sugar diet, the flies seem to be eating more, not because they're always hungry, but actually it's their meals are getting longer. And the reason their meals are getting longer and bigger compared to the controlled diet here in the salmon color seems to be because they can get satiated by the meals as in the diet without the added sugar. And so I'm not going to talk about this today, but if you're interested in it, feel free to ask me any questions afterwards. The papers where you can read about this are here. So how does diet shape taste, sensation, and feeding behavior? So to answer this question, we must be able to measure taste, sensation in flies. And so to do this, we use an assay called the proboscis extension response, or PER, where the fly chemosensory neurons, the taste neurons, are stimulated with a sugar solution, like in the cartoon, and the fly will extend its proboscis to eat the food. And now we can measure and quantify this behavior where no extension is a 0 and a full extension is a 1. And so we can essentially measure the intensity and the sensitivity of the fly to sweetness. So when we do this and we give flies in a controlled diet, which in the talk I've showed with a banana, and we give them things that are barely sweet like zucchini and things that are really sweet like soda, you see that flies have this taste reactivity curve where they respond more with proboscis extension to things that are more sweet. And then when we put them on a sugar diet here with a cupcake for 7 days, we see that there is between a 20 and 50% decrease in their ability to taste sweet. And this is not because they can extend their proboscis, they can still taste everything else. This is specific to sweetness. And in fact, we can see this phenotype developing as early as 3 days on a high sugar diet, here shown in the red line. So we know that when we give the flies a high sugar diet, their sense of sweet taste is blunted, that we've shown here with a great cupcake. And using different measurements of neural activity and responsivity, such as calcium imaging, sensory recording, and vesicular release, we essentially found that flies don't taste sweet as sweet because their taste neurons don't work as well. And what we also found is that this blunting or dulling of sweet taste actually is what drove this bigger meals and increase in feeding behavior and eventually the fat accumulation. And we showed this in different ways, but one critical way was to essentially use closed-loop optogenetics to stimulate the taste neurons only when the fly was eating the food, and so to give them a virtual experience of sweetness. And when that happened here in the pink line, and we're measuring their feeding behavior over different days on a high sugar diet, we see that their feeding behavior remains stable compared to the control flies, which are escalating. And so the pink flies now have normal, regular meal sizes, and they remain lean. And so we know that the taste changes are actually pretty important in the development of obesity. So this really gave rise to 2 different questions. One question is, how do changes in taste modulate eating? How do we go from changes in taste to food intake? And then the other one is, what is about a sugar diet that's actually changing the sensory neurons? And these two questions are essentially the questions I talked about before in my cartoon. Now, today I'm only going to focus on this one. And if you're interested of hearing about how changes in taste modulate feeding behavior and meal size, we just published a paper authored by Christina and 3 different undergraduate students in eLife last month. So I encourage you to read about that if you're interested in that, and that you can reach out to me, and I'm happy to answer any questions, or Christina. So what drives changes in sweet sensory neuron activity? And this is a thesis project of Anumit Vaziri, who's an incredibly amazing and talented young woman who in the next year and a half is going to look for postdocs in Europe, I hear. So make sure you reach out and connect with her, because she's the real deal and you definitely want her in a lab. All right, so and before trying to get to mechanism, we actually decided to do some dietary manipulation to try to figure out what really was causing, what about the diet was causing the taste changes. So I'm also going to summarize all this work with one slide and cartoons. But essentially, we first took flies that were genetically had obesity, but these flies could taste just fine. Then we gave flies a lard diet, which has the same amount of calories as the high sugar diet. And this diet, the flies also accumulate fat, but they could taste sugar just fine. We also gave flies a diet of high sweetness, so kind of diet soda, and this also didn't have any effect, so they could taste just fine. It was only when we gave fruit flies a diet of glucose, fructose, or sucrose, even in flies that were genetically lean that didn't accumulate any fat, that we were able to induce this dulling or this chemosensory plasticity. And so around the same time, Daniel Walensky, a postdoc in the lab, and a former technician, Jasmine Winsler, had done this metabolomic study to look at how metabolites and nutrients change with a high sugar diet. And that's also published, so I'm not really going to talk about it. But in this particular study, we were able to identify a metabolite called UDP-glucnac and a pathway, a metabolic pathway called the exosamine biosynthesis pathway, as being essentially increased by a high sugar diet. And in fact, a lot of other people had found this already, so that was not surprising. But what was surprising is that this pathway actually was both necessary and sufficient to mediate the plasticity of the taste neurons. And so I like to think of the exosamine biosynthesis pathway as the mTOR of sugar. It senses different levels of sugar rather than different amino acids. But in fact, it's really tuned in, not just with glucose metabolism, but a lot of different nutrient sensing pathways in the cells. Now, at the end of this exosamine biosynthesis pathway, you get a metabolite, UDP-glucnac, that's used not for energy, but it's used as a signaling, as a moiety to add on proteins as a post-translational modification, very much like phosphorylation, just we're using a amino sugar moiety for that. And the enzyme that does this job is O-gluconic transferase, or OGT. So in our paper, in our work, we had found that OGT essentially is mediating the effects of a high sugar diet on the physiology of the sweet sensing neurons in a cell autonomous way. And so we were then interested in trying to figure out, well, what's downstream of OGT? Because this gives us a handle to try to understand that. It turns out that OGT modifies, post-translationally modified, a lot of things in the cells. And so since people had identified with these things, we did a bias screen where we looked at the ability of things that had been known to interact with OGT to be necessary and sufficient for sweet taste, chemoplasticity, and response to diet. So what Anumit found during her rotation, actually, she identified the polygroup group proteins are targets or are being involved in chemosensory plasticity. So what are polygroup Polycomb group proteins. They are perhaps one of the most well-known and most ancient pathways involved in gene repression, and they do this through histone modifications. They're conserved from plants to humans, and they mediate gene silencing via histone 3 lysine 27 methylation or histone 2A lysine 119 ubiquitination. And the cool thing is that PRC1 and PRC2, the polycomb repressive complex 2 and 1, their activity is both positively modulated by the OGT metabolic sugar enzyme I just spoke to you about. So what Anomi found is actually only the polycomb repressive complex 2, which I'm going to refer as PRC2, is involved in sweet taste deficits in response to diet. So PRC2 is made up by this protein called EZ, which is the methyltransferase, and two obligate accessory components without which the complex just fall apart. And then if we now test this mutants on the proboscis extension reflex to measure fly taste, here's the control flies in the control diet with the dots. And on a high sugar diet, we see that the fly sweet taste sensation is decreasing in response to different concentration of sucrose. But in the PRC2 mutants shown here, the sweet taste deficits are prevented or almost entirely prevented. And notice that on a controlled diet, these mutants have largely normal taste. Now, PRC2 actually is pretty helpless without associating with proteins that brings it to chromatin. It cannot really do much. And so it needs something called recruiters. And there are different recruiters that bring it to chromatin. And so Anomi tested many different of them. And she found that only the polycomb-like protein, PCL here in pink, was necessary to prevent sweet taste deficits compared to control flies. And not only did Anomi find that PRC2 mutants were required for sweet taste deficits in response to a high sugar diet, but also that if we did pharmacological inhibition of PRC2 by using an inhibitor of ESC, remember I told you that those two subunits are required or the complex falls apart, then with the inhibitor right here, the flies on a sugar diet, the squares, did not experience taste deficits compared to the control flies plus the vehicle on the sugar diet right here. So it seems like PRC2 is necessary for sweet taste deficits to develop in response to a high sugar diet. So Anomi wanted to know if this is actually happening in the sensory neurons. So to do this, she used a driver that only expresses in the sweet tasting cells to knock down PCL only in the sensory neurons. And remember, PCL is a recruitment component. And so because it's what's recruiting the PRC2 to diet, it's the best genetic manipulation to do to actually influence the activity of the complex, and the most specific because PRC2 comes in different flavors. So in pink here are the experimental flies, and in black are the flies on a high sugar diet, the control flies, and knockdown of PCL in the high sugar diet also completely rescue sweet taste deficits. And the knockdown here was about 50%, and we have different RNAi lines. And interestingly, if you do the opposite experiments where now you're overexpressing PCL on a control diet, now we can induce taste deficits even in the absence of the high sugar environment. And so this is really interesting for a couple of reasons, and I put a star there so I will remind myself. First, both with the RNAi and the mutants, we don't see any, in order to see the phenotype, we have to put the flies on the diet. And so this is a typical gene by environment effect where the flies can taste fine, the mutant flies or the RNAi can taste fine on a controlled diet. It's only when we put them on a high sugar diet that we can rescue the phenotype. So one possibility is that Polycomb-like and PRC2 are actually just altering the number of sweet sensing neurons. So we label the sweet sensing neurons here with GFP. You can see all these beautiful sensory neurons in green. And both when we knock down PRC2, when we knock down PCL or overexpress it, we saw no changes in the number of sweet sensing cells. So it cannot just be the cell number. So before I told you that we had seen changes in neural responses to sucrose with a high sugar diet. So we went back to doing this experiment, which was done by Christina May before she moved to NYU for her postdoc, where essentially we are expressing calcium sensitive green fluorescent protein only in the presynaptic terminal of the sugar sensing neurons. We open the fibrine, we stimulate the fly proboscis with sugar, and then we look for changes in fluorescence as a proxy for neural activity. And so what we find in the control flies right here on a sugar diet, you see that the responses to sucrose are lower compared to the control diet. But now if you look at the PCL mutants over here in pink, the control and high sugar diet are essentially the same. So PCL mutants are able not only to rescue the taste behavior, but also to rescue the responses of the taste neurons to sucrose. So just let me summarize this for a second. So far I've told you that the Polycomb repressive complex 2 modulate chemosensory plasticity on a controlled diet, on a high sugar diet here. Control flies experience a decrease in their ability to perceive sweetness, which is reflected in the lower responsivity of their sensory neurons. But if we use PRC2.1 mutants, knockdown or inhibitor, we can completely eliminate this phenotype, and if we overexpress PRC2, we can induce this phenotype. Now, if the flies are not experiencing a change in sweet taste, and as I told you before, changes in sweet taste are important to modulate feeding behavior, then we would expect that this fly should also not experience diet-induced obesity. And in fact, when now we look at the level of triglycerides, of how much fat they're accumulating after 7 days on a high sugar diet, the controls accumulate quite a bit of fat, whereas our PCL knockdown just in the sweet sensing neurons, the flies remain lean and have the same essentially body fat as control flies in a controlled diet. So I gave them a party hat because they can eat all the sugar they want and they don't have to worry about potential negative consequences of it. So I also told you earlier that we got to PRC2 via OGT by looking at things that OGT potentially could modify. So do OGT and PRC2 work in concert, or are they working in different pathways? Now, this line of work was severely impaired by the 4 months of shutdown. So Animate wasn't really able to do any experiments. But what I can tell you is that we have pretty strong genetic evidence that essentially is placing OGT upstream of PRC2 PCL. We do not know if this interaction is direct, but now that we're back in the lab, Animate can start answering these questions. And so for today, I'm actually going to focus on trying to figure out how PRC2 and PCL are actually reshaping and sculpting the physiology of the sweet sensing neurons in response to a high dietary sugar. So to answer this question, we really needed to know whether PRC2.1 binding actually changes with a high sugar diet. And it turns out that there's a technique called TADA that can do this. So in TADA, exploits the ability of a DNA adenosine methyltransferase to essentially methylate DNA at specific sequences. And now we can take the domain of this DNA adenosine methyltransferase DAM and fuse it to our favorite chromatin or DNA interacting protein. And now we can use DNA methylation of the DNA as a proxy for essentially binding or occupancy of our favorite protein. So Anomate made DAM PCL transgenic flies. And as a control for this, you also have to look at essentially where the DAM methylase by itself is going, because there's always some background DNA methylation. But it also turns out that you can exploit this if you keep the methylation at bay, you can actually exploit this to measure the accessible chromatin, because the DAM will only go to the pieces of the chromatin that are accessible, that are not highly compacted. So we actually did this induction of both of these flies between the third and the fourth day on a control diet and on a high sugar diet. And our reasoning was that we really wanted to see how things were getting started, look at the transition rather than the endpoint at 7 days. And I told you before that we already see detectable phenotype after 3 days of a high sugar diet. So because this technique has not really been used a lot before, especially in sensory neurons or small population of neurons, we did a lot of QC, and I'm only going to cover a few of it here in the interest of time, but I forgot to mention that this manuscript is on bioRxiv, and so we have a lot of supplemental figures to essentially show that this technique is working well. But first, we can look at the accessibility of the DAM in our neurons. And we would only expect the DAM to be present in the sweet taste receptors, but not in the bitter taste receptor, because they're expressed in different cells, if we actually are able to target the right cells. And in fact, we can see an increase of accessibility in all the 3 different reps, GR5A, but not in the bitter receptors. And we also did quite a little bit of work to see where PCL was binding in the chromatin of the sweet taste neurons in a controlled diet, and we found that in fact it was binding in a lot of the canonical targets of the Polycomb group proteins, which you can see here in this big cluster of 500 kb. And you can also see the characteristic sort of binding of the Polycomb proteins, which is kind of not a nice transcription factor, but they're kind of coding the chromatin. And interestingly, if you look at the accessibility of this locus here, then you see that the catada by itself cannot get there because the chromatin is very compacted. And so this gave us confidence to then look at the control and a high sugar diet. And as I said, we only pulse the methylase for about a day between days 3 and day 4. Now, where does PCL and PRC2 bind on a sugar diet and a control diet? So it turns out that the PRC2 PCL complex doesn't just bind anywhere, but there are specific 6 regulatory regions in the DNA to which it binds that I put in pink here. They're called PREs, for Polycomb responsive elements. And if we now look at the PCL binding at the PREs, we see that it's much increased with a high sugar diet here in purple compared to the control diet in gray. And then if we look at the accessibility of this PREs on a sugar diet, it's much decreased compared to the control diet. And so on a high sugar diet, PCL is occupying PREs at a higher level, and the chromatin accessibility is lower. Now, when we look at what changes between a control and a high sugar diet, we found about 400 to 450 peaks to which Polycomb-like was changing between the 2 diets. But to our surprise, we didn't find a complete increase in binding. In fact, we found 2 different groups. In one group right here, we found that PCL had increased binding on a high sugar diet, but we also found another group where PCL had decreased binding on a high sugar diet. So there is a redistribution of PCL binding rather than just an increase in binding everywhere in the genome. And then when we did GO-term analysis, pathway enrichment analysis to see where it bound, to see what the genes it was bound to were doing, essentially 95% of them are all regulatory. So they're transcription factors, and some of them are involved in transcription repressor activity, neuroblast fate commitment, axon target recognition, and proboscis development. And so we know that the direct targets of PRC2 on a high sugar diet are actually transcription factors. We didn't find any channels or receptors. There were a few metabolic enzymes, but for the large majority of them were transcription factors. So the next question, now that we know that, is how does PRC2 redistribution actually reshape sweet taste? How do we go from this redistribution of chromatin to an actual phenotype on the ability of the fly to taste? So to ask this question, we use another technique called ribosomal profiling, and we use flies that were developed by Dion Dickman Lab. And actually, Dion was so kind to share the flies with us before they were published. And so at the time, this was also not really used a lot. And in the paper, you can find a lot of QC for it. The technique works pretty well, and I think this is perhaps the best indication of it if we're looking at the enrichment that we get for our sweet sensing neurons. Oh, here's Cupcake. Hey, girl. Sweet sensing neurons, which are labeled by the gustatory receptor 5A. So in the sweet sensing neurons here, you can see an enrichment for the sweet taste receptors and also for the fat taste receptors that is expressed in the same cells. But there's no enrichment for beta receptors like 66A and 32A. So we did this ribosomal profiling experiments both at 3 and 7 days on a high sugar diet because we wanted, again, to look at the cells in transition to losing their taste and towards the end where the phenotype is very strong. And our DAM-ID experiments are sort of in between these two. And so the first thing we noticed is that on a high sugar diet, actually, both at 3 days and 7 days, we see a negative skew in the fold changes in gene expression. So almost 90% of genes are down regulated here, and about 68% of genes are 3 days. And this is particularly obvious. Oh, and if we now use PCL mutants, we can actually not only reverse this negative skew, but even overcompensate it. The ability of PRC2 to really affect the transcriptional response to a high sugar diet is really can really be seen in this heat map. So I know heat maps are not great, especially for tox, but bear with me for a second. Everything that's yellow or gold goes down. Everything that's purple goes up on a high sugar diet. And there are about 700 to 800 genes at 3 days, 700 to 800 genes at 7 days, and about 200 of them in common. Now, for the sake of time, let's just look at the sugar diet 7 here. You can see most of the genes here are down. They have negative log to fold changes. And now just a few of them are increased here in purple. And then when we mutate PCL and we put the flies on a sugar diet, then you see that essentially nearly all the changes that were caused by dietary sugar are reversed or not change anymore by mutation PCL. So PCL seems to be really a master regulator in reshaping the transcriptional responses of these neurons to diet. And when we do the pathway enrichment analysis, we find that most of these genes are involved in synapses, signaling, metabolism, and transcription. And of course, the transcription really caught our attention because remember, from our DAM-ID data, we found that PCL binds to transcription factors. So what we did, we cross-reference our chromatin binding with our ribosomal profiling. And first what we found is that the genes that are bound by PCL on average have lower transcription, lower expression or RNA abundance than the genes that are not bound by PCL. And we 5 of the genes particular caught our attention. They are 5 different transcription factors. 4 of them here are activators, and 1 here, scarecrow, scrow, is a repressor. Now, these are the track for binding of PCL on a controlled diet in black, on a high sugar diet in purple, and the difference in cyan between the control and a high sugar diet to the chromatin of the sweet sensing neurons. And so you can see here there is an increase in binding of this 4 loci on a sugar diet, and there is a decrease in binding at the scarecrow locus. This redistribution that I was thinking about. Now, when we look at the RNA levels now here, but in our ribosomal profiling data, what we found is that scarecrow actually levels, mRNA level increase consistent with the decrease in chromatin occupancy, whereas the level of GATA-E, NUB, PTX1, and CAD decrease compare to a control diet, consistent with the increased occupancy of PRC2 here. And then in PCL mutants, this was completely reversed. So we got really interested in these 4 transcription factors because it potentially gives us now another entry to try to understand how these transcriptional responses are shaped. And so just to summarize, so far I've showed you that this nutrient-dependent redistribution of PRC2 occupancy on the chromatin of GR5A neuron shift the balance of transcriptional regulation in the taste neurons. On a control diet, PRC2 is sitting on top of scarecrow, and these other 4 transcriptional factors are expressed. And on a sugar diet now, PRC2 shifts to repress transcription of this 4 regulator, and SCRO is expressed. And we know we're interested in this because we know that something about this is related to the ability of the cells to respond to sucrose, which ultimately affects sweet taste sensation, which, as I showed you before, affects the ability, the metabolic state of the fly. And so we wanted to understand how we go through this shift in transcription, shift in balance of regulators to changing activity of the cell. And so we collaborated with Peter Fredolino, who's computational biology at the University of Michigan. And what Peter and his postdoc Morteza Kabiri did is to essentially tile the entire fly genome with the consensus binding sites of this 5 transcription factor that I just spoke about, a 1 base pair resolution, to essentially determine which are the potential targets of these 5 transcription factors. And so then once we identify this candidate targets, we can ask how are they behaving on a high sugar diet? And so in here, I'm showing the 5 different transcription factors. 4 of them here with the green street light are activators, whereas SCRO is a repressor. And if you look at the log2 fold changes of, oh, there's a typo here, sorry. If you look at the log2 fold changes on between a control and nice sugar diet, you see that they're all negative. The targets of CAD and everybody else are all down. And then when we do the same analysis on our PCL mutant flies on a sugar diet, we're able to reverse all of them. And this is really interesting, especially to think about the fact that the genes that are repressed by PCL are activator, and the gene that's de-repressed by PCL is a repressor. Because remember, I told you that by 7 days, almost 90% of all the gene changes are negative. So there is a massive transcription repression. And so this could explain why we see such a strong effect. Now, if these transcription factors are really important, we would expect them to have an effect on sweet taste. Ultimately, what we care is about behavior. I mean, I love chromatin, I love transcription factors, they're my favorite, but ultimately behavior is my more favorite. And so can we modulate some aspect of sweet taste function by trying to genetically modify the levels of this transcription factor as if the fly was on a high sugar diet? And so what we did here, we used RNAi just in the sweet sensing neurons for each of the 4 activators, and compared to the control diet flies, these are their taste reactivity curves, we see a decrease in sweet taste on a control diet. So all these transcription factors are necessary for sweet taste. On the other hand, if we overexpress scro control diet, we can induce taste deficits. And so that's extremely cool, because we can go from a change in chromatin and RNA abundance to a change in behavior by trying to essentially recreate the same conditions that a high sugar diet recreates. However, when we try to do the opposite experiments and overexpress these four activators on a sugar diet to see if we could now rescue sweet taste deficit, we found that it didn't work. So they're necessary, but they're not sufficient. And so we started scratching our heads, and we asked Peter to essentially look at the overlap between the different targets that he had identified for each of this transcription factor. And what we found is that actually there's quite a lot of overlap. The GATA E is perhaps not surprising, given the nature of this transcriptional regulator, but it is really interesting to see that SCRO actually is inhibiting some of the same targets that PTX, CATNUP, and GATA are stimulating. And so this is probably an explanation for why each of this transcription factor, when we overexpress the activators, it's not sufficient, because SCRO, the SCRO negative repressive drive very likely is still there. The other thing that we noticed is that a lot of this are developmental genes. And so developmental genes a lot of times have this transcriptional relationship within each other, where they either stimulate their own expression or stimulate the expression of partners for which they bind at enhancers. And so when we did a computational analysis of the target, we in fact found such feed-forward relationship between the activators that I'm showing here. Well, that Ani actually drew this, that Ani is showing here with the green arrow. And then you can see SCRO is actually computationally directly inhibiting GATA-E and CAD. And so this is probably how PCL, by controlling the 5 of them, can create such strong repressive drive across the cell. And it's very upstream, and it can really drive the transcriptional responses to a high sugar diet. The other thing that's interesting is that looking at the pathway analysis for these genes, although there is overlap, they seem to also possibly control different aspects of the physiology of the neurons. And so here we did go-term analysis of the targets of each of these transcription factors. And for example, PTX1 and CAD seems to be perhaps more involved in perhaps the genes that would modulate the sensilla responses, the dendritic responses to the sugar detection, whereas things like NUP and GATA-E might be important for more synaptic changes, like synaptic vesicle release, synaptic assembly, and terminal boutons. And so we're really excited about this. And a postdoc in the lab, Hayan Sung, is starting to essentially trying to understand how now we go from this transcriptional and chromatin changes that we know have an effect on behavior to actually linking them back to the physiology of the neuron. So what I've told you today is that we have identified a new epigenetic mechanism through which diet can modulate neural state and change behavior and disease risk. And this new mechanism involves the Polycomb repressive complex 2 together with the recruiter Polycomb-like. And we identified this essentially transcriptional hub that's involved in taste plasticity. And I didn't have time to tell you about, but we identified genes that are specifically required for taste function downstream of it. So you can look at the paper on bioRxiv and stay tuned for Hayan's work. And that this changes activity. And so you might say, well, Monica, you wrote epigenetic, but you really didn't show us anything epigenetic, right? I haven't showed you anything related to histone modification. Everything I've shown is chromatin binding and RNA abundance changes. And you're absolutely right. And we cannot actually do those experiments. They're probably technically close to impossible right now. But a hallmark of epigenetic is persistence, right? And so we can ask, are the sweet taste deficits persistent? And in fact, this is what everybody always asks us at the end of the talks. And so I'm going to spend the next few minutes just in a few slides telling you whether they're persistent or not. So yes, taste deficits persist. And so to do this, Anumid, together with MNT undergraduate student Brandon Janab, what they did is to take the flies that had been on a sugar diet here in purple and put them on a controlled diet for 7 days. Recently, Anumid has extended this up to 20 days. And so these are our controlled diet to controlled diet flies. They can taste sugar. They live a happy life. These are our control diet to sugar diet flies, everything I've been telling you about before. Their taste decreases. And now if we take these flies and we move them back to a controlled diet for 7 days, they, here in green, their taste doesn't change. So their sweet taste deficits are locked in. And they're locked in for about 20 days. That's as far as we've gone. But that's, you know, pretty long, almost 50% of a fly life. So to try to figure out how this was happening, Anumid repeated the ribosomal profiling experiments comparing this reversal flies to the control diet. And in fact, she found that there's still a negative skew in gene expression. And it's particularly interesting to compare here where I have the red arrow, these sugar diet to control diet flies, to the sugar diet 7 flies and the other flies I've been talking about, the other conditions. And you see that they actually cluster with each other. So the flies that have been on a controlled diet for 7 days are more similar than the flies that have been on a sugar diet for 7 days than any other conditions. And you can see here that genes still are this gold color, which means that there's still transcription repression. And in fact, when we look at the entire transcriptome, we find that about 55%, a little over half of the transcriptional change mediated by high sugar diet compared to the controlled diet are sort of locked in, irrespective of diet. But we can find a way to actually get rid of them. And that is by using the inhibitor of PRC2 I spoke early on, where we can dull the activity of PRC2. So when I told you before, we use the inhibitor in the context of a sugar diet. If we give the inhibitor of PRC2 on a sugar diet, we can rescue sweet taste deficits, which you can see here. These flies can taste as well as control flies compared to the sugar flies without the inhibitor. But then we can do another experiment and we can inhibit PRC2 just when the flies are on the controlled diet right here. And when we do this, now we can rescue taste compared to this persistent effects we described and I just told you about, which means that PRC2 is required for the persistence, and we think it's actually constitutively required. So this is why we think that this is an epigenetic mechanism, because there is a persistent component that locks the cells into this state. And there is another, I think, really exciting idea that comes up of Anumid work, and it's the idea that developmental programs might be re-engaged to modulate plasticity. In fact, I told you that this is one of the classic developmental programs in flies, but what I haven't told you about is that most of the genes that are repressed, these transcriptional activators, are actually enriched in the sweet sensing neurons compared to the rest of the brain, whereas SCRO is actually enriched in the central brain compared to the periphery. And so we think the sugar diet to change the physiology of the neuron is actually re-engaging this developmental program. And so this kind of piqued our interest because it suggests that perhaps the idea of cell fate, which we know Polycomb proteins are so important for, and the idea of cell state, which as neuroscientists we think of plasticity, perhaps are connected. And this is something that we're really excited and interested in the lab. So to summarize, I've shown you today that the sensory neuroscience, the sensory system, and how it's changed by nutrition, it's really important for health. In flies, through the work of many people in my lab, we have been able to causally link changes in taste to changes in food intake and the metabolic state of the animal. And we are starting to do some of this work in rats. But there are a lot of different labs, and I'm sorry if I forgot to list them in here. There are a lot of different labs working from flies to rodents to humans that have really witnessed different aspects of this feed forward a vicious cycle of diet and feeding. And so I think there's accumulating evidence from flies to mammals and humans that components of processed food like salt, sugar, and fat can reprogram our sensory system to promote eating and increase the risk for metabolic disease. And I feel this is something we should study in order to perhaps one day put forward regulation and guidelines, because as you might know, there are actually no guidelines for sugar content in food because sugar is considered generally recognized as safe from the FDA. And so essentially manufacturer can choose how much sugar to put in food or how much salt. Okay, so I want to thank you for your attention. I already spoke about the people that did the work. Anomid Vaziri did almost all the experiments with the exception of one. She's a really incredible scientist, and do reach out to her if you're interested in talking to her more. And Brenda Janelle was the undergraduate student that Anomid talked to, that Anomid mentee for the Persistent project. I also want to thank you all the people that shared reagents with us and our past and current collaborators, and you for your attention.
Okay, thank you so much, Monica. It was a wonderful talk. Okay, so we have quite some few questions over here. Okay, so the first question, can we have a control where the diet has sweetness but no calorie to rule out that ATP is not modulating this? Also a control with proper osmolarity of the diet?
So we did the... Oh, so my headphones are dying. Let me just switch the audio. Let me just switch the audio. Okay.
Oh, Monica, we cannot hear you anymore. We still cannot hear you, Monica. Maybe, was it that your mic was connected to your... I see. How do you want to... Do you want to... if you want, you can... write your questions in the chat, maybe. I need to type on Monica's screen. Okay,
right.
Someone is saying that you are muted. Someone was saying that you are muted. No,
yeah,
we hear you.
It just took a minute.
We hear you again.
It just took a minute.
Okay,
yeah,
we
just
have you again.
For this okay, so we did do an experiment where we gave flies only a high diet. Oh, Zita, can you
Oh, sorry.
So we give them a high sucralose diet, So same calories as the control diet, but high sweetness, and we didn't see any effects on taste. So we didn't see any dulling. In terms of osmolarity, we gave flies water, agar, a little cup with agar, together with the high sugar diet, and we also didn't see any effects of that. We also seen effects on the dulling of taste with concentration pretty much 15% and above. So we have 15, I think 20, 23, and 30. So even barely above what would be maybe most flies diet are 8%, we see taste changes.
Cool. Okay. So next question, really cool talk. Could you explain a little bit more about what is upstream of PRC2-PCL? Is its expression activity dependent? And if so, do you have an idea of how increased activity in the taste neuron on a high sugar diet affects PRC2-PCL?
So we don't see any changes in PRC2 levels, RNA-wise. We think that our working model is that OGT is modifying the activity of PRC2. Now, this is being shown during development to be the case, and we know that in the absence of OGT, PRC2 overexpression is not sufficient to induce taste deficits, which means that OGT is epistatic to PRC2. So OGT is before PRC2. We have some ideas of what OGT would be doing. OGT is known to actually modify different Polycomb proteins. And so one way could be by changing EZ activity, which has been described, or another way it could change PCL activity. And we're now trying to figure out. But we think that OGT activity is higher because the levels of the metabolite, the UDP glucanac, is higher on a high sugar diet. And OGT activity is linear across all level of cellular UDP glucanac levels. And so the more UDP glucanac you have, the more OGT would be able to modulate or post-translationally modified proteins. So stay tuned for that.
Okay, so the next question. Sorry for the naive question, but I did not fully understood the behavioral response to high sugar diet. Is there a lower extension of the proboscis, and this means lower sweet intensity perception?
Yes, so the behavior is measuring the extension of the proboscis, which in the case of sugar measure the intensity of which the fly is experiencing the sugar. So it's either a no extension or a decrease in extension depending on the fly, likely depending on how many cells in the proboscis are damaged at the point where we're taking the measurements, because there are about 50 to 60 sweet sensing cells. Now, it's not that the fly cannot move the proboscis, it can eat other things and move it, but it's that the high sugar diet is decreasing the responsiveness of the sensory neurons. So when they feel sugar, they're not as stimulated by it. And we showed recently they cannot even transmit the signal as potently as before. So the signal that the brain that it's processing the sweetness flavor is even weakened.
Great. Okay, so we have another question here. Does PCL overexpression in the sugar sensing neurons, which decreases the PR, also affects physiology and leads to the overweight flies?
We haven't tried those experiments yet. We're close now to trying the overexpression, and our prediction would be that it does. Now, for the high sugar diet, for the obesity, what we found is that a fly has to be able to experience the change I don't think the change is conscious, but the fly has to experience the change in order for the chemosensory plasticity to affect food intake. So, for example, if you take a fly and silence the sweet sensing neurons, then from birth, so that the flies never gets to experience the beautiful sweetness of a banana, then it's there is no effect. So the fly has to experience it, and then to experience sweetness, have the sweetness decrease, and then it will have an effect. The chemosensory plasticity has an effect. And so it's really the plasticity of the change that matters rather than the absolute output of the sensory neurons. And we have some speculation why that is. We think it's because of how central brain circuits are then processing that change. And we don't think it actually has something to do with the liking that the fly has for the food, mostly based on human data, but actually with how the fly is using the sensory cue of the sweetness as a way to modulate expectations about the food. And so the Eli paper I just spoke about was about part of this, and then Thibaut Pardo, another graduate student in the lab, is actually studying how feeding expectations and towards food, well, how expectation and memory towards food are changed by a high sugar diet because of changes in taste. So kind of a complicated answer, but we don't think it's the liking. We think it's the way the cue of sweetness is being used.
Cool. Okay, so we have another question here. So the question is whether the high sugar diet kind of just causes some damage overall to the nervous system of the fly, whether the mutant or the wild type situation.
Well, we don't think it's causing a damage because we can rescue it. So it's not like a persistent damage, and we don't see any major changes in like gross changes in the anatomy. We don't see any changes in the number. A lot of times, if you'll see the shape of the cell body would be different. So we don't see any cells dying, and the mutants are actually rescuing, right? The mutants are making the taste better, not worse. So we don't have any evidence with the methods we have employed that it's damage. But I think, what do you mean with damage? Do you mean like the cell dying? We don't see that. But to me, there could be a change in connectivity, very likely. I mean, a very hard thing to measure. There could be changes in the number of synapses. And so I think it depends what you mean with damage. But if with damage, you mean change in connectivity or the number of synapses, the way synapses are wired, they talk to each other, I think it's highly possible. And I think our data suggests that's the case, at least from the GO-term analysis.
We have a couple more questions. Maybe we can answer them if we are quick. So one of them is, besides the taste neurons, do you have information about taste cells? Are they totally function and the changes are only at neuronal level? I don't know.
Well, I don't know if this person is asking about glia cells that are in...
Yeah, I don't think it's clear what would be the difference between taste cells or taste neurons, right? Because it's kind of okay.
So I think I know. In taste in mammals, there are taste cells, and there are sensory neurons that carry the information from the taste buds to the brain. And in flies, it's a simpler system where the taste cells are neurons. So I think that's where the question is coming. So So in mammals, actually, we've recorded from the sensory neurons of rats, and other people have, and we see a 50% decrease in the responsivity to sucrose. With a high sucrose diet, essentially the same as here. Other people have given rats or mice a high-fat diet and a high-fat, high-sugar diet. And one group, when they give them a high-fat diet, they actually see a change in a decrease in the turnover of the taste buds, but other people with a high sugar diet don't see it. And that group actually thinks is the metabolic state. So when a mouse has really high obesity and metabolic syndrome, then there's something going on to the stem cells. And a lot of other people have described the stem cell changes with obesity. Okay, I guess.
Okay, exactly. Okay, so the last final question. Do we know the half-life of your proteins of interest, those five of them?
No, I don't. And certainly we wouldn't really be able to address that question in the sensory neurons.
Okay. Thank you so much, Monica, for your talk.
Thank you.
It was a wonderful talk. So for the ones who joined for the discussion, let's just meet maybe in 5 minutes or so in the Zoom room. Thank you so much. See you.
All right. Thank you, everybody. Bye-bye.
Thank you, Monica. See you in a couple of minutes.
Yep. Bye.